Hello, my name is Taylor Smith. I will be presenting with Shane Lawson and Ian Truebridge about the signaling pathways of the mitochondrial unfolded protein response. In lecture, we learned about the unfolded protein response in the endoplasmic reticulum. To summarize briefly, there are different transmembrane kinases that ultimately recruit chaperones to assist in protein folding. We will go into mechanistic detail shortly, but the gist of the unfolded protein response in the mitochondria involves an accumulation of a transcription factor, ATFS1, in the nucleus in response to mitochondrial stress. ATFS1 upregulates mitochondrial chaperones while simultaneously limiting transcripts for the TCA cycle and oxidative phosphorylation. This is so that the mitochondria can focus on achieving proteostasis. Our topic of interest in this presentation involves the investigation into another pathway in the mitochondrial unfolded protein response, which operates in conjunction with ATFS1. In this study, the mitochondrial UPR was investigated in the worm C. elegans.
As mentioned previously, one pathway is mediated by ATFS1, which upregulates mitochondrial chaperones to attend to the abundance of unfolded protein. The other pathway is mediated by the kinase GCN2 and the cytosolic transcription initiation factor EIF2-alpha. Both methods for dealing with mitochondrial stress, the upregulation of mitochondrial chaperones and the downregulation of cytosolic translation, occur in parallel pathways. The researchers discovered that the loss of function of a single pathway places more stress on the complementary pathway. The group hypothesized that the loss of function of both pathways is more detrimental than the loss of function of a single pathway.
As other studies have shown, mitochondrial dysfunction triggers the HAF1-dependent accumulation of ATFS1 in the nucleus, resulting in the upregulation of mitochondrial chaperone genes, including HSP60 and mitochondrial HSP70. Activation of this pathway occurs in response to mitochondrial stress, which results from the accumulation of unfolded proteins beyond the capacity of normally present mitochondrial molecular chaperones.
GCN2 is a kinase that phosphorylates EIF2-alpha, which is a cytosolic translation initiation factor. Phosphorylation levels of EIF2-alpha increase during mitochondrial stress, which uses reactive oxygen species that are generated from dysfunctional mitochondria. Although GCN2 is a crucial component for functioning during mitochondrial stress, experiments have shown that GCN2 deletion lines can still function and live in the absence of stress.
This study showed that the GCN2 and ATFS1 pathways work in parallel while under mitochondrial stress to maintain protein homeostasis. Additionally, they showed that GCN2 was required for development and lifespan extension while under mitochondrial stress. Manipulation of GCN2-dependent EIF2-alpha phosphorylation may have therapeutic applications for mitochondrial dysfunction-related diseases. This can be accomplished by reducing cytosolic amino acid levels or small molecule inhibition. Thank you for listening to our presentation, and have a great day.